Thank you so much for joining this session. I am Nina Prater. I'm a soil specialist with NCAT in our Southeast office in Fayetteville, Arkansas. And I am so excited to be here with Dr. Buzz Kloot. Dr. Kloot is a research associate professor in the Environmental Health Sciences Department at the University of South Carolina. And I got to hear Dr. Kloot present at the ATTRA SARE conference a couple years ago in St. Louis, Missouri. And I just really enjoyed his talk on the HANI test. He was going over the research findings on that. And I thought it was so inspiring. And I just really appreciated how he looked at soil testing as a tool for farmers to use to improve their soil health, and just looking at different approaches and what we can take from those and apply them and move towards a better soil health outcomes. So I'm really excited about this talk. Just a few housekeeping things. It's being recorded. Please enter your questions for Dr. Kloot in the chat box at the bottom of the Zoom screen, and I'll ask those at the end. And then if you are getting CEUs, the QR code will pop up at the end of the talk at the last few minutes, and you can do your QR code magic with that. I don't even know how that works, but I'm sure you do at this point. And then please rate this session at the end on the Whova platform, so let us know how we did. So with that, I'll turn it over to Dr. Kloot.
All right, thanks very much. When people say Dr. Kloot, I kind of look over my shoulder because
who's that?
Nobody calls me Dr. Kloot over here. Everybody calls me Buzz. So anyway, hi guys. Good to talk to you. It's nice, you know, while I would love to be in Montana right now, it's really nice that I can do this from my lab. And really, I can't remember what the name of the talk was, but really what we wanted to just do was kind of look at some of the practical issues of soil tests, especially biological soil tests. We did a little bit of this work last year, and I think what I see a soil test is it's an important, but it's a very imperfect window into the state of the soil. And so what we're going to review today is looking at 4 different kinds of soil tests that'll include physical, chemical, and biological components. We're going to look at 3 soils. One is degraded, one is in progress of being regenerated, and one is very regenerative. And then in our part of the world, we're going to look at the 2 ecosystems, or 2 ecosystems. One is the Piedmont, which tends to be a fairly shallow but loamy clay soil. So if the moisture is there, it's very fertile. Whereas our coastal plain soils tend to be a little bit sandy, but much deeper. So that's the contrast there. And then I'm going to give you a few thoughts and observations on these soil tests. The first soil that we're going to look at is actually in my rose garden over here. About 10 years ago, this was basically pure clay covered with moss and lichen. And people used to use it to drive up and down to do construction on. We bought the piece of land, and I started to cover it first with mulch and then later on with cover crops. And what you see right now is my roses and then some turmeric plants that I harvested in October this year. So this is probably one of the most fungal soils that I've seen under the microscope. So that's, you know, we're going to say it's a regenerative soil. It's an Orangeburg sandy loam urban complex. So it's got a lot of building material when you start digging around below the A horizon. But typically it's a loamy sand. And when you actually look at the soil structure, you know, for the top 6 to 12 inches, all you're going to see is this BB, you know, like BBs or cottage cheese soil structure, the granular soil structure. The second soil is a pasture in the Piedmont. These are some of my friends from Clemson. And this was a cotton field, you know, continuous cotton for many, many years up to the north of Columbia, South Carolina. Sandy clay loam and clay loam soil. My friend over here, Kneeling, went ahead and started to graze this. So it's a perennial pasture and uses annuals to regenerate it. So we're seeing improvements in the soil. We've seen improvements in the soil over the last 3 or 4 years, but it was very degraded. And the last one is a degraded coastal plain soil. It's a Norfolk sand. And you can see here was our first attempt to cover crop the soil. And basically it's been soybeans and collards for the last 20 years irrigated. And so it's fairly well degraded over here. So that's kind of a contrast of the kinds of soils that we'll be looking at today. And over here, I wanted to show you there are 4 soil test labs that we used. We used Cornell, Clemson, Ward Labs, and then Waters Labs. For those of you who don't know, Waters Labs is a lab run in Georgia. It's a commercial lab. I often use them for some of my consulting just because they turn stuff around really quickly. So if we look, all 4 of the labs do some kind of chemistry. They go ahead, but they have different extraction methods, as you can see. So Cornell would have the Morgan. Clemson does Malic 1, which is probably quite uncommon. Usually Clemson and Georgia, the land-grant universities still use Malic 1. North Carolina and Florida use Malic 3. So it's just a different way of extracting the chemicals. And the idea is to try and simulate what's happening around the roots. We'll talk a little bit about Ward and how what kind of methods they use as an extraction method. Cornell also does an aggregate stability test. I'm not sure if we actually display those, but Waters also does an aggregate stability test based on the Solvita method. Cornell looks at soil carbon, both or percentage organic matter. They look at a 96-hour carbon dioxide test, and then they look at active carbon. I think there's a specific nickname they give it, and it escapes me right now, but they use potassium permanganate there. Ward uses, looks at organic matter loss on ignition. They look at the water extractable organic carbon. That is a term that Rick HANI has used. And essentially it's a sample that where you run a certain volume through a filter of water through a certain amount of soil, and you measure the carbon in the filtrate. And then over here, Ward uses the 24-hour CO2C method over here. I don't know about Cornell, but Ward uses a particular carbon dioxide sensor that the HANI labs used. Waters also does organic matter in terms of loss of ignition. And sort of a good rule of thumb that we use is 58% of organic matter is carbon. Now, you know, some people say it's exactly 58%. I think that really varies, but at least, you know, you can say 2/3 of or just less than 2/3 of organic matter is going to be composed of carbon. They also do the 24-hour CO2C method, but they use the Solvita method over here to to look at their soils. So it's a Solvita colorimetric paddle that they use. And you can actually use those as well. In terms of soil nitrates, or not nitrates, nitrogen. So one of the biggest things we've had in the last few years is that we're not just looking at nitrates or ammonium, the inorganic nitrogen, we're looking at a number of other species as well. So in terms of Cornell, they use the autoclave citrate or ACE soil protein. So they're looking at soil protein. My understanding is the soil protein, you're often going to find that in the membranes of microbes that really, as those microbes get consumed or get eaten up, that protein then gets made available to the plant roots. Clemson usually only does the nitrate N, and typically they use that for their pre-side dress nitrate tests. Ward, again, uses the HANI standards, water extractable organic nitrogen, and then they also compare that to the inorganic nitrogen species, namely ammonium nitrogen and nitrate nitrogen. Waters kind of does the same thing, ammonium and nitrate nitrogen, but they look at the Solvita label organic nitrogen. I'm just going to minimize this because actually I can't see that last screen, that last thing on the biological soil tests. So bear with me a second here. Okay, so in the biology, really the one that I wanted to share with you was the one that Ward does. They use the phospholipid fatty acid test. Now, I'm not saying that that is the be all and end all of all biological tests. In my lab, I use a $700 microscope where we can get a good estimate of bacteria, protozoa, fungi, nematodes, and we can quantify that to some extent. But the PLFA is convenient for us because I don't always have students or grad students who are willing to spend half an hour to 45 minutes looking down a microscope for each sample. So that kind of gives you an idea. And so what we'll do is sort of walk through the soil chemistry first. And I'm going to point out the light blue over here is Cornell. The orangish is Clemson. The green bar is Ward. And then the purple bar is Waters. And so if we go to the first sample site that we took, that was my garden. And you can see it's blowing up with carbon. And I find this really amusing. I know Cornell sent me a note and said, please stop adding manure and phosphorus to your soils because they're dangerously high in phosphorus. Well, the funny thing is I have never added any kind of manure or phosphorus to the soil. So I don't know what happened in the soil biology, but it is bursting with soil test P. And so I can't explain it. All I can say is it must be the biology because it sure, it was basically used as a driveway and parking lot for the last 40 to 50 years. So that's the soil test phosphorus that you can see for the first site that we have. The second site was this soil, and you can see, 4 and 5 parts per million of soil test P. Remember, we're using different extractants, so we're not expecting the same numbers. But what's interesting, or what I would like you to note, is that there's a general trend towards, if this one is ranked number 1, then the others are also, the others tend to be ranked in the same order. So you can see this one has the most phosphorus here. Interestingly, the degraded coastal plain soil also has a fair amount of phosphorus. It wasn't picked up that much in the Cornell sample here. But you can see more or less the rankings are the same. So again, we have to, I guess I'm always a little bit careful not to get hung up on specific values, although, you know, in fertilizer recommendations, they have soil test ranges. And we'll talk about that in a little while later. Interestingly, soil test potassium, again, I'm going to have to just slip out because I can't see. In this case, soil test potassium in the rose garden is the highest. In the pasture, where we've got a lot of clay, you can see that's next up on the list. And not surprisingly, in the coastal plain soil, it's the lowest. Again, we're using different extractant methods for each one of them. Let's move on to soil carbon. And I guess you wouldn't be surprised that carbon and things like, you know, potassium, especially other cations like I find usually carbon and calcium are often very, very well related. And over here we can see, I think all of them used a similar method for carbon or organic matter. And the rose garden, again, not surprisingly, had a fairly high organic matter. The pasture over here, it's getting better and better. Also has, for our part of the world, in the Piedmont, an organic matter of above 2% in some of those degraded soils is on its way to being mended. And then the third one, the degraded coastal plain soil, I was surprised to see that we even had as much as 1.5% in the Clemson, the Ward analyses. And I don't know if that might be because we'd started with cover crop. The second way we measure carbon is this respiration method. That's the 24-hour respiration. And essentially, it's a method of indirectly measuring what the activity is in the soil. So I'm going to just escape again because I can't see the last one. But you can see there's, in the rose garden, a terrific amount of soil test respiration going on in the rose garden. Remember, the Cornell is a 96-hour one. The Ward one is a 24-hour one, as is the Waters. The difference is Waters is using a Solvita paddle, and I think often, I think this has been rectified, but Solvita at one stage, they will, because the paddle gets put in a container of a limited volume, they were saturating out at about 105 ppm. So I suspect this value is a little bit higher. If you can have a look where we don't have as much respiration, the values are much closer. And over here, in fact, the values are slightly reversed. Again, these are biological values. And so biology doesn't always behave like we want it to. But again, what you can see is this kind of ranking from highest to lowest over there with our respiration. If we have a look at the third measure of soil carbon, we have the active potassium permanganate carbon. In this one, in the second one under the Ward labs, the green one, that's your total organic carbon, and that's carbon that is in the pore space. So it's water extractable organic carbon. And then finally you have that, this is Waters. They're also using the active potassium permanganate carbon. So you can see Waters and, whoops, let's go back, excuse me. Waters and Cornell numbers look fairly close. So they're looking at this, I guess, soil protein or proteins in the soil. Oh, sorry, that's carbon, excuse me. So they're looking at that particular amount of carbon, whereas HANI is looking at the carbon in the pore space or the dissolved carbon. A rule of thumb that HANI usually has is that there's about an 80th to 100th amount of carbon in the spore space compared to the carbon in the organic matter. And I find that to be generally true. So again, different ways of measuring carbon in the soil. When we look at soil inorganic nitrogen, we're just looking at nitrates and ammonium. And interestingly enough, you can see the producer in the coastal plain soil. He was putting a lot of nitrate fertilizer out there. You can see that. But what we do see is, at least in my experience, is that our soil test ammonium generally kind of never really exceeds much around 5 parts per million. So it tends to be a lot less variable that we can compare to our soil inorganic nitrates or our soil nitrates. So a lot more variability in nitrates, especially as a function of, when it was last fertilized, with some kind of commercial fertilizer. Soil organic nitrogen. So over here, we've got 3 different types. We've got the ACE protein, that's from Cornell. We've got the water extractable organic nitrogen, again, from the HANI test that Ward does. And then we've got this Solvita labile amino nitrogen. So Solvita and Cornell are looking at maybe something similar, but their methods are different. Again, you can see the ranks for each one of these. If you take each test individually, you'll see the ranks follow one another. And that's kind of one of the points that I wanted to make here, just about for most of these things. In other words, part of this is no single soil test is going to be your salvation, or it's going to somehow improve your soils on its own. You've got to follow those soil health principles, keep the soil covered, keep a live root in the soil year round, minimal disturbance and diversity. And we usually like to add the one of incorporating livestock into that. So remember, soil tests are not going to get you soil health on their own. They're just a window, and you've got to be aware of the pros and cons of each soil test. Let's quickly walk you through the soil microbe array. We've got bacteria and fungi. And in this slide, this is on my high school microscope I have in my lab over here. Typically what you see, come on now, where's my pointer? Okay, here's the pointer. What you can see is some of these little transparent spots over here are probably going to be bacteria. This is probably a particle of quartz, and some of these particles might be clay particles, but these little transparent spots that float around typically are going to be some of the bacteria that we're going to find. This is a fine specimen over here of a saprophytic fungus. It's dark. It's about 3 microns or more in width. It's got, and you can't really see it, but it's got septa. In other words, it's got very distinct blocky cell walls that separate it. And this classes it as a quote unquote good guy fungus. Often in commercial agriculture, anything that's microbial is considered a bad guy. And by the way, we have something to kill it for you. Well, hopefully we're reversing that perception to say, well, if we've got a good balanced system where we're following the soil health principles, you're going to see a lot more good guys than bad guys. So this is a good guy fungus over here. So if we look at our phospholipid fatty acid, not surprisingly, we've got a boatload of bacteria in my rose garden, a little less in the pasture, and even less in the in the degraded coastal plain soil. So you not quite an order of magnitude, but in the rose garden, about 3 times more than in the pasture, and about 5 times more than in the coastal plain soil. In terms of the fungi, sorry, I have to look again. You can see in this case, in the rose garden, we've got an order of magnitude or more better than the coastal plain soil. Remember, they're both the same soil texture. They're a loamy sand or a sand, whereas this is a clay. So typically you'd find soils are more, clay soils are going to be a little bit more protective than that. But it's phenomenal to see how much variability you have in that. The other thing I wanted to measure or mention is that in a disturbed system, because your soil bacteria are much smaller, they will hold out a lot longer than your soil fungi. So when you go in and put fungicides on the soil and slice and dice with tillage equipment, your fungi are some of the first things to go because those fungal hyphae get pulled apart and exposed to air, and they will oxidize and be preyed upon by opportunistic microbes. Let's just look very briefly at some classes of fungi. Again, my high school microscope. This fungus is a saprophytic fungus, so it gets most of its energy from consuming lignin, below the plant. So a lot of plant roots are lignin, and then, your residue is also lignin. If you look under a pile of residue, often you find those white hyphae. That would be a classic case of soil saprophytic fungi. This is a lovely picture. I was so excited to get this again with my high school microscope. We got some soil roots from a friend, some corn roots from a friend in some regenerative soil. This over here is a root hair. And what we did was we cleared it with a caustic mix, and then we stained it with a blue stain. And so you can look up clearing and staining if you do have a microscope. And what we found was these fungal hyphae that have formed a relationship. They have essentially germinated inside the plant hairs, and you can see they're much skinnier than the plant hairs. And I'll bet you $1 that these were much longer before we went ahead and cleared and stained it. But you can see these mycorrhizal fungi are actually living in symbiosis with this plant hair, and they're the guys that are going out and picking up nutrients, and they maybe even have symbiotic relationships with bacteria and other fungi that are able to secrete things like phosphatase or mine little soil particles to get exactly the right nutrient they want. So what do these guys get in exchange for their services of bringing water and nutrients to the plant? Well, they get carbon in exchange, right? So the plant photosynthesizes, and when it photosynthesizes, it makes sugars, and these guys pick it up and then also give carbon to their partners out in the soil. So these are the saprophytes. Their carbon is dead carbon. These saprophytes, their carbon is sugar directly from the plant. So the phospholipid fatty acid method actually uses, we can actually see that. PLFA, it actually looks at the phospholipid fatty acids in the different microbe cell walls. I forgot to explain that to you. Again, over here, you find a lot fewer saprophytes as you go down from the very regenerated soil to the less regenerated. And then you'll also find over here your mycorrhizal fungi are much, much higher. So again, you can see that fungal to bacterial ratio. I think I look at it in a little bit, but the fungal to bacterial ratio typically is much, much higher in that sandy soil. You might have heard that bacteria and fungi, people often refer to them as little bags of fertilizer. Well, little bags of fertilizer are no good if you don't have fertilizer bag openers and spreaders. And this is a tested amoeba. So we also have little creatures that we generally call protozoans. So we have amoeba, we have ciliates, and we have flagellates that run around. And if you have a look, they're consuming these bacteria at a phenomenal rate. And then they're pooping, they're consuming it, and then they're pooping out, ammonium and nitrates after they've consumed these critters. And all of this is happening right next to the plant roots. So these guys are, and the nematodes are really responsible for the recycling of nutrients. Again, this is my high school microscope over here. So if I can do it, anyone can do it. So over here, what we can see is lots more protozoans. In other words, we've got fertilizer bag openers and spreaders over here, and they are recycling those nutrients at a frightening rate. And maybe one of the explanations of why we have such high phosphorus, according to Cornell, dangerously high phosphorus levels in my rose garden that I never fertilized. And that's probably because I've got these protozoans and they don't measure the nematodes, but these guys are running around and consuming the bacteria and fungi as fast as their little paddles can carry them. You can see we've got some protozoan or predator activity in Don's field over here, but we've got nothing here because it's a disturbed soil. They practice tillage. I mean, lots and lots of tillage. So there are no protozoans, there are very few fungi, very few protozoans, so that's no habitat. In terms of the fungal to bacterial ratio, you can see this is pretty good, 0.29, 0.23, and I imagine the one in the degraded soil is low, sort of less than 0.01 over here. So let's look at, one of the maxims I use, and I remember making some people mad when I used it, and then they came back to me later on and said, oh, I get it right now. I think you need to understand that all soil tests are wrong, but some are useful. So don't get me wrong. I have a little consulting company and I have a lab, and I use all manner of soil tests. But I never assume that any single soil test is the answer to the question. So just use that maxim. All soil tests are wrong and some are useful. So one way to interpret soil tests, well, let's have a look first over here. There is an agronomist by the name of Brad Jorn, Dr. Brad Jorn. He was a professor of agronomy at Purdue, and he looked at the average pricing for a 2-year cycle of corn and soybeans, 180 bushel corn, 60 bushel soybeans, and looked specifically at a specific soil test level in each one of these states. And he came back with the fertilizer recommendation for each state for this amount of corn and soybeans and for a given soil test level. And then he assumed 50 cents a pound for nitrogen, phosphorus, and potassium. So obviously there are some basic assumptions. But Brad pointed out the disparity between states, even though we've got the same amount, the same yield over here, the disparity between state fertilizer recommendations, not just between, say, Vermont and Illinois, but take a look at the difference between Vermont and I think New Hampshire. It's about a 2.5-fold difference. Look at the difference between Indiana and Illinois. You know, Brad was saying, well, what if you're a farmer on the border? Do you believe the Indiana recommendations or the Illinois recommendations? And I'd say the same thing if you have a look down here in the bottom, you're looking at Georgia and Alabama. You know, you're looking almost over a 2-year cycle of $100,000 difference in just fertilizer recommendations. So we need to understand that fertilizer recommendations certainly have their place, but we need to be very, very careful of people defending their fertilizer recommendations as the standard, and you know, it's repeatable all over the country. Clearly, this is not the case, and I wanted to make that point that Dr. John made quite a few years ago. He was looking at the phosphorus index, by the way, and this was part of his argument. So let's move on. I also wanted to talk about the HANI test, just a few words. How long have I got, Nina?
Just maybe 3 more minutes, and then we'll
oh my goodness. All right, 3 more minutes. Okay. So HANI test, just remember, basically it's got 3 parts. Not only the soil, the respiration, it's also got the water extraction part. And then finally the H3A extract, where they use organic acids to extract rather than inorganics. So remember, it's not a single number, it's a bunch of numbers over there. I'm going to move on. Over here, what I wanted to point out to you was my friend and I grew a cover crop over here, but we took a 20 by 20 foot strip over here and killed the cover crop in November. This was taken in early March. And then we measured the soil test, and we saw where we had cover crops, we had significantly higher total biomass, bacteria, rhizobia, and fungal biomass, and then also our vascular mycorrhizal fungal biomass. So we could actually objectively see, and this was an average of 5 plots, how our cover crops actually improved or enhanced the biology. The other way of soil tests that we use is we like to track things like organic matter and soil pH. This happens to be a field in my part of the world. The maxim has always been, if you don't apply lime every 2 years, your soil is going to acidify and you're going to lose your crop. Well, this one we've had from November 13 to December 18 and longer, or it was a series of 13 fields that we had once we started cover cropping, our soil test levels leveled out. Organic matter, what we've noticed in the same fields is with cover cropping, we seem to be increasing soil organic matter at the rate of 0.05% per annum, which is about 1,000 pounds per acre. So the last word before I open it up to questions is, you know, just for you, when you're looking at soil tests, decide what you want to observe, chemical, physical, chemical or physical. Do you want to look at soil carbon and nitrogen, and do you want to look at the biology? Settle in on a lab or the labs that you feel you can trust, and don't jump around, because if you go from one lab to another, you're not really going to get good historical data. Also remember, there's more than one way to interpret the soil test. And just because a lab says this is our way doesn't mean that it is the only way. And last thing I'd like to leave with you is, remember, all soil tests are wrong, but some are useful. And I'm going to leave it to Nina, and we'll go from there.
Thank you so much. That was really interesting. So I'll just jump right into some questions we got in the chat. What type of soil tests could potentially act as a broad baseline data set to support a watershed soil health program, something that gives quantitative that is a quantitative means to track management changes and look at trends over time?
If you're looking at managing a watershed, and I might be old-fashioned, but I still think there's a lot of value in just tracking the soil chemistry over time. Also, just because it's so darn cheap, and looking at organic matter, we do see that organic matter increases over time. You know, we've got several experimental farm fields, because we don't have a at my university, we're not a land grant university, so we don't have any experimental farms. So I work with farmers, but typically we're seeing that increase. But the other thing that if you can do this is now you're actually able to look at soil stability and soil aggregate stability test. And I would also recommend that you look at soil bulk density and infiltration rates. So these are not difficult, these are not expensive, but they do require a lot of field work. And if you take a soil bulk density, you know, take 4 or 5 at the same place so that you have a good average. And I have infiltration rings. I have 4 infiltration rings that I'll always put in because infiltration is highly varied. But honestly, still, those would be the ones, if you're trying to stay within a budget, those would be the ones that I would look at in terms of watershed management.
Great. You mentioned the aggregate stability test.
Yeah. Yeah.
Is that something that people send their samples off to a lab to do, or is it something you could do on your own?
Right.
What's the best way to do that?
Yeah, well, you know, that's a question I have for one of my graduate students. I'm still waiting for the answer. The answer is that you can actually do it. I think Ornada, if you go to ornada.com or .net, that's HORNADA, they have a method, but again, it's kind of lab intensive. Solvita Labs actually has a method that I think they will certify you to do. And then I know Cornell does that. Solvita will do it in their lab for you as well, but Solvita will sell you the equipment. So I think if you look around, you can find something. So again, I don't know what's best. For us right now, we're looking at aggregate stability because often the soil chemical test or the soil organic matter test doesn't always emulate what we can see with our eyes and feel with our hands. So I would, and then so the ag stability test at least helps us quantify that. So we're kind of balancing against sending it off to a lab for sort of results that are just a number versus maybe getting it into our lab and then getting some experience of interpreting that data.
Mm-hmm. Okay, we have a question from my colleague in California, Rex. He says there's a lot of seasonal differences between soil tests from one location. How often would you suggest to do soil testing to understand changes in soil organic matter and fungal to bacterial ratios?
Yeah, that's a super question. Okay, first of all, I have been talking about this for years and years, and in my consulting business this year, instead of looking out the window, I sent my guys out to sample in November and December when it was raining cats and dogs. And we had cold saturated soils, and guess what happened to our soil chemistry? It really tanked. And you know, we're going out and we're going to have to take a few repeat samples there. So in terms of your soil chemistry and soil pH, it's always good to take samples at the same time each year. I have traditionally taken those samples in mid to late fall in South Carolina, that's November. But this year I made the mistake of taking it when we had excessive rainfall. So those numbers were meaningful. What I have noticed though is organic matter, and the old-fashioned loss of ignition organic matter for us seems to have stayed quite steady. Now, if you were to ask me what the best time would be to take a sample, it would be right before planting. Right before planting everything is greened up. and you have your soil biology is really kicking over and going crazy. Sometimes your cover crops are head high, and that's when I would take it. For example, we've seen with PLFAs, where we have a straight up no-till versus a cover crop system. We've got 3 or 4 times more biology under the cover crop, and then we grow corn for 100 days, and then we go back to those exact same sites, and we can see no difference between the PLFAs. So very quickly, that changes. But I would recommend, if logistics allow, just before you plant. But often, a lot of guys can't do that because the labs are clogged up. So a lot of guys often do that just after they harvest. And there are pros and cons to each one of them. And the one reason why we don't do it straight after harvest here is we've still got another about 60 days of growth. It's often very, very hot, at least after the corn harvest. And it's a fantastic way to sell lime, because after corn is harvested, our pHs are often about a point lower than 2 months later. So the soils actually get to neutralize over that time. And, there's anecdotal evidence that in our case, that producers were encouraged to have their samples taken so that lime sales could go up. That's kind of taking a cynical view, but I think the point is that sampling should be seasonal. Be aware of, try and do it at the same time of the year, but be very aware of, are your weather conditions typical, in terms of soil temperature and soil moisture.
Right. You may have just answered this, but someone else asked, given the changes in soil moisture and microbiology, is it better to tie a soil test to a date or a phenological state of a crop if you want to track soil carbon and organic matter?
Okay. I would say, yeah, ooh, hello.
We're almost done.
Can I get credits for my own? I don't think I've got a single credit this year.
I think that works.
All right. I would probably tie it to the crop growth stage or the cover crop growth stage. But, I would still want to be in the window of that date. And, possibly the other component there would be soil moisture and temperature, kind of that's where I would cruise towards. Because remember, if you're taking a soil test and you're doing a crop rotation, you're not going to be comparing apples to apples if you're going, say, I'm doing row crops here, corn and soybeans or cotton or something like that. So maybe soil temperature as well. Soil temperature and moisture might also be a good part to do that on.
Okay, great. Well, we're almost out of time. And so I think that'll be the last question. There are a few more in the chat that we can try and get to after the conference. I just wanted to thank you for your statement of all soil tests are wrong, but some are useful, because it has helped me help other farmers who sometimes get analysis paralysis with the soil tests, and they want to get everything perfect. And it's really helped me help explain to them the way to use a soil test. So I wanted to thank you for reiterating that today, and for all the great information that you shared. And it's been a real pleasure.
Well, thank you so much, Nina. Nice to meet you again.